Hello everyone, welcome to Molecura. Today we are going to be discussing glycolysis in details, and we are going to see what happens during each reaction and why do they happen and how do they happen. So we are now going to talk about glycolysis and how does each reaction happen. In glycolysis, we are starting the process with one molecule of glucose and we're finishing it with two pyruvates. At the end result is going to be two molecules of pyruvate. Now there are 10 reactions that happen in glycolysis until the glucose that we have here gets converted to 2 mole of pyruvate. And we're going to go through the reactions one by one, and then we will look at each reaction to see what is happening in the structure of that molecule and what is getting converted to what exactly. Now the first reaction is going to be the conversion of glucose to glucose 6-phosphate. So this reaction here is going to give us glucose 6-phosphate. Now the phosphate group is going to be attached to the carbon number 6, and that's why it's called 6-phosphate. So our phosphate group is going to be attached to this place. Now where does this phosphate group come from? It comes from hydrolysis of one molecule of ATP. So basically here we are using ATP to keep this reaction going, and this is important because once we add the phosphate group to the molecule of glucose, this glucose cannot get out of the cell, so it will commit this molecule of glucose inside the cell. Now it can be used for glycolysis or it could be used for other pathways, but the main point here is that this molecule of glucose is now committed to that particular cell, and because the phosphate group has a negative charge to it, this molecule cannot freely get out of the plasma membrane and get out of the cell. Now this reaction is going to happen by the enzyme that we call hexokinase. So the enzyme is called hexokinase. Now what does kinase mean? Kinase means enzyme that is adding a phosphate group using ATP. It's not always like that, but that's the case most of the time. So what's happening here? A molecule of ATP is getting consumed and it's getting converted to ATP. So in the first step, we actually consume ATP instead of producing it. And the additional phosphate group that gets attached here is come to the carbon number 6 of glucose, and it's going to give us glucose 6-phosphate. Now the name of this enzyme is important to remember because this enzyme is regulated. Most of the cells have hexokinase in it, but the same enzyme, when it's inside the liver, we call it glucokinase. On the next video, we're going to talk about the regulation of glycolysis, and it's going to be an important topic to discuss for medical biochemistry as well. So now it's important to take a look to see what is happening on the molecular level. So here we're going to look at the same reaction again. One molecule of glucose is getting converted. If you can see here in this molecule, we're going to have 6 carbon atoms. So this is the first one, second, third, fourth, and fifth. And here we have the 6th one. And on the 6th one, we are having an addition of a phosphate group. This is our phosphate group. And again, why is this important? Because we don't want this molecule of glucose to get out of the cell. So this is our phosphate group that's getting attached to carbon number 6. Now, as we said, this is happening through the action of hexokinase. I'm just showing the reaction in 2 different ways, so it might be easier to grasp and easier to understand. And one ATP is getting consumed here, and it's getting converted to ADP. So this molecule that we have here is now called glucose 6-phosphate. Why? Again, because it has a phosphate group on carbon number 6. So this is our first reaction of glycolysis. So the second reaction in glycolysis is going to be creating an isomer out of the glucose that we have. Now, this glucose 6-phosphate needs to be changed in its configuration. So our enzyme here is going to be called phosphoglucose isomerase. So it's isomerizing our glucose that has a phosphate group in it. So the name shows that. The name is phosphoglucose isomerase. Some texts would just refer to it as isomerase. So this is going to convert our glucose 6-phosphate into fructose 6-phosphate. And this is particularly important because once this 6-carbon molecule gets broken, we need it to be in a different form, on a different configuration for the rest of the glycolysis to go ahead. Now, we're going to have a look at this to see what is exactly happening. So here we have it. Here we have our glucose 6-phosphate. This is our glucose 6-phosphate. And you can see again that our phosphate group is on the 6th carbon. So this is getting converted to fructose 6-phosphate through the action of our enzymes that we said is called phosphoglucose isomerase. Now, in this step, it's important to note what is happening exactly. So here we have a carbonyl group. This is what we call a carbonyl group. This is our carbonyl group. If a carbonyl group is at the end of a carbon chain, it's going to be called an aldehyde. So our glucose technically is called an aldehyde. But now this aldehyde is getting converted to something we call a keto. Now, if our carbonyl group is in the middle of the chain, like it is here, this is our carbonyl group. This is our carbonyl group. If it's in the middle of the chain, it's going to be called a keto group. It's going to make fructose a keto sugar. So we are changing the chemical configuration of glucose and we are making fructose 6-phosphate. So what we have here is called fructose. It's called fructose 6-phosphate. And again, the phosphate group that we have is going to be on carbon number 6. So this is carbon number 6. That's why it's called fructose 6-phosphate. So this is our second reaction, glucose 6-phosphate. It's getting converted into its isomer, which is fructose 6-phosphate through the action of phosphoglucose isomerase. And glucose, which is an aldehyde, because of this carbonyl group, is getting converted to a keto because the carbonyl group now is in the middle of the chain. Now we are going to the third reaction of glycolysis, which is one of the most important steps. Why? Because this step involves an enzyme which is highly regulated in the body. And we are going to talk about this in the next video, which we are talking about regulation of glycolysis. And this enzyme gets regulated in different ways that we need to know about it. So what does this enzyme do? This enzyme is a kinase. So it's adding another phosphate group to our glucose 6-phosphate. So far we had the glucose 6-phosphate, which was here, and it has a phosphate group in it on carbon number 6. So now we are going to have an addition of another phosphate group, which comes on carbon number 1. So now our molecule is going to have 2 phosphate groups. And this enzyme that does this reaction is called phosphofructokinase. The enzyme, let's write it here. So this enzyme is called phosphofructokinase 1. Again, we are having a phosphate being added to our fructose 6-phosphate. And this phosphate has to come from somewhere. And again, it's going to come from ATP. So one ATP is getting consumed again. and it's getting converted to ADP. And what we will have as a result is our molecule that now has 2 phosphate group on it. What would this be called? We would call this molecule fructose 1,6-bis phosphate. Now, the naming is important, okay? So what it's telling us? It's telling us that what we have here is a fructose. That we already know. We had a fructose here, so now we are also having a fructose. This fructose has 2 phosphate groups on it. It's on carbon number 1 and it's on carbon number 6. Carbon number 1 and carbon number 6. And the phosphate is shown here. So fructose 1,6-bisphosphate. So why do we use bis to describe this? Because the 2 phosphate groups are not next to each other. So we are not using di or di to describe this because the phosphate groups are apart. In the previous video, we showed that adenosine D-phosphate is called D-phosphate because the 2 phosphate groups are next to each other. Here, the phosphate groups are far apart. That's why we use bisphosphate instead. Now, we want to look at this to see what's happening on the molecular level. Here on the molecular level, we're going to have our fructose 6-phosphate. That's getting converted to fructose 1,6-bisphosphate. Now, this is happening, as we said, through the action of the enzyme that's called let's write it, the whole thing, phosphofructokinase 1. We have a phosphofructokinase 2, which we're going to talk about it in the next video. And here it is important to know what's happening. Every reaction, when it's happening in biochemistry, you can easily tell what is getting converted to what by looking at the name of the enzyme. Here we have fructose with a phosphate group. We are adding a phosphate group to it. So it's going to be a kinase. The kinase is adding a phosphate group to what? To the phosphofructose that we already have. So where does this phosphate group come from? It's going to come from ATP, which gets hydrolyzed and consumed, and it's going to be converted into ADP. So this is the second reaction of glycolysis, which ATP is getting consumed. In the later steps, ATP gets produced, and we're going to have a net production of 2 ATP, as we're going to see. So now we look at fructose 1,6-bisphosphate to see what is happening exactly. We can see here that we have again carbon number 1, carbon number 2, carbon number 3, carbon number 4, 5, and 6. We have a phosphate group here. This is our phosphate group on carbon number 1 and a phosphate group on carbon number 6. That's why this is called fructose 1,6-bisphosphate. And as you can see, we already talked about this. This structure is different from glucose because its carbonyl group, which is this, the carbonyl group is in the middle. The carbonyl group is in the middle of the chain instead of being top here, which would make an aldehyde. So this is the third and one of the most important reactions of glycolysis. Now we will go to the fourth reaction. So in the next reaction of glycolysis, we have something we call an aldol cleavage. Aldol cleavage is what's happening in this reaction. As we can see here, this molecule that has 6 carbons is going to be cut into half, and half of it will be here and half of it will be here. And this is called an aldol cleavage. We're going to talk about when we see this structure in a moment. So this enzyme that's going to catalyze this is called aldolase. So this reaction is happening over here. It's going to this side and it's going to this side. Now, the enzyme that's responsible here is going to be called aldolase. And what's doing? It's doing an aldol cleavage. And we're going to have two molecules here, okay? One of them is going to have a phosphate group on carbon number 1. The other one is going to have a phosphate group on carbon number 6. One of these is going to be called dihydroxy acetone phosphate. And the other one is going to be called glyceraldehyde 3 phosphate. Now the glycolysis pathway is going to continue using glyceraldehyde 3 phosphate. So our dihydroxyacetone phosphate needs to get isomerized into this molecule, and it's going to happen through the action of an enzyme that's called triose phosphate isomerase. That's called triose phosphate isomerase. Triose phosphate isomerase is going to isomerize DHAP, which is dihydroxyacetone phosphate, into glyceraldehyde 3 phosphate. And in the remaining reactions of glycolysis, we are going to have two molecules of glyceraldehyde 3 phosphate continuing the pathway. So from here onwards, each reaction is happening twice. Why? Because our fructose 1,6-bisphosphate got cut into two, and one of them became glyceraldehyde 3 phosphate and one of them became DHAP, and the DHAP is getting isomerized back into this molecule here. So now we are having two of the same molecule continuing the glycolytic pathway. Now, to look at it on a molecular level, we can see what is happening here. It's the same thing. It's going to start with an aldolase, and our aldolase is going to convert our fructose 1,6-bisphosphate into two molecules, and our two molecules here are going to be called DHAP and G3P, which we're going to write here in more details. So the enzyme here is called aldolase, and what this enzyme does is an aldol cleavage that's happens here. We call this an aldol cleavage that happens here, and it's going to give us two molecules. This molecule here is going to be called dihydroxy acetone phosphate. And this molecule here is going to be called glyceraldehyde 3 phosphate. So this can be called G3P, and that's what we will write on our main page, and this is going to be called DHAP. Now, there is an isomerization reaction that's happening between the two, and it's going to be through an enzyme that's called, and this could happen both ways, by the way, so we can write it as both ways. It's called triose phosphate isomerase. Now, if we can see here one more time, we are going to have an aldol cleavage, and we are going to have two molecules. One is here, one is here. We have already talked about the names, and you can see here the carbonyl group is in form of an aldehyde, and here the carbonyl group is in form of a ketone group. And what happens is that this gets isomerized into glyceraldehyde 3 phosphate. So from here onwards, we are going to have two molecules of glyceraldehyde 3 phosphate that continue the glycolytic pathway. So the next reaction in glycolysis is going to be an important one, again, because a lot of things are happening here. Now, here we have a molecule of glyceraldehyde 3 phosphate. So it is important to note that for each 1 mole of glucose that enters glycolysis, by this stage we are going to have 2 mole of glyceraldehyde 3 phosphate. So any ATP production we have from here onwards is going to be happening 2 times. Now, this reaction where glyceraldehyde 3-phosphate gets converted into a molecule that we call 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate. So what is happening here is, as the name suggests, we had a phosphate group here. Let's draw it downwards so we have space for all the reactions that's happening. And we had 1 on the carbon number 3. We had a phosphate group. Now on carbon number 1, we are also adding a phosphate group. So 2 phosphate group is now on this molecule. So what is the enzyme that is catalyzing this reaction? The enzyme here is going to be called glyceraldehyde 3-phosphate dehydrogenase. Is going to be called glyceraldehyde 3-phosphate dehydrogenase. And the dehydrogenase enzymes are going to be catalyzing a redox reaction, a reduction oxidation reaction. What is getting oxidized in this reaction? In this reaction that we have over here, our glyceraldehyde 3-phosphate is getting oxidized, which means it's losing electrons. We have explained this in the previous video when we talked about the redox reactions. So the oxidation is happening to glyceraldehyde 3-phosphate. In other words, it's losing electrons. So where do these electrons go? These electrons are going to go to NAD+, which is our electron carrier, and it's going to get converted into NADH. So in this reaction, we are having glyceraldehyde 3-phosphate getting oxidized, which means it's losing electrons. And these electrons are going to the NAD+, which is getting reduced, means it's gaining electron. And this electron is going to convert NAD+ into NADH. We also have an addition of an inorganic phosphate that's coming into this reaction. So we have also an inorganic phosphate that is getting into this reaction to make our 1,3-bisphosphoglycerate molecule. So this is an important reaction because at this point we are having generation of our first NADH in this glycolytic pathway. And it's important to note that since this reaction is happening twice, we'll have 2 molecules of NADH getting produced during glycolysis. And in case of anaerobic respiration, which means there is no oxygen, no TCA cycle happens, no electron transport chain happening, we need to regenerate this NAD+ to be able to keep the glycolytic pathway from going on. We will talk about this in detail when we talk about anaerobic respiration. Now, let's look at this from the molecular level to see what is happening exactly. This point of view is particularly important because we are looking at the specific bonds that are happening in this reaction. Here we have our glyceraldehyde 3-phosphate, and this is our carbonyl group. This is our carbonyl group. Now, what is happening here, and we talked about the name of the enzyme. Let's try to write it down here as well. The enzyme that's working on this reaction is called glyceraldehyde 3-phosphate dehydrogenase. And we said that this is going to be a redox reaction, which means that we are having an NAD+ in the reaction. Our NAD+ is going to accept the electron, which the glyceraldehyde 3-phosphate is losing. It will accept the electron and it's going to get reduced to NADH. And we're going to have an addition of an inorganic phosphate coming into this reaction. So this comes from the cytoplasm, not from an ADP. And that is important to note, and that's why the enzyme is not called, for example, a kinase. Now, it's important to note in this reaction that we are having something that's called an acyl phosphate group. So it's important to note here, what we need to know is an acyl phosphate is getting added here. So this bond is going to be a high energy bond. So this happens when a phosphate group gets added to a carbonyl group, okay? So this is our carbonyl group. Our carbonyl group is always going to be like this. So our carbonyl group is here, and we have a phosphate group getting added to it. And what we have here is going to be a high energy phosphate bond. So this is going to be, that we have shown in purple, this is an high energy bond. So this bond in the next reactions is going to get hydrolyzed into ATP, and our ATP production, the first ATP production that we're going to have in glycolysis, is by breakage of this bond and transferring this phosphate group to ADP to form ATP, which we're going to see in the next step. Now, to conclude this step, we need to look at the naming of this molecule here. What is this molecule is going to be called? So we have two phosphate groups. One is on carbon number 1, one is on carbon number 3. So what we have is 1,3 bis phosphoglycerate. 1,3 bis phosphoglycerate. Why? Because our phosphate is on carbon number 1 and on carbon number 2. Bis, why? Because the two phosphate groups are away from each other. Phosphoglycerate, which is going to be the name of the molecule that's there. Now we're going to look at the next reaction of glycolysis. So this reaction is going to be very straightforward. We're going to have our 1,3 bisphosphoglycerate getting converted into 3-phosphoglycerate. So what's happening is that this phosphate here is getting removed. It's going to go to ADP to form ATP. So the reaction here is going to be through an enzyme that you might have guessed the name is going to be called phosphoglycerate kinase. And kinase, why? Because it is transferring a phosphate group into something, and here it's ATP. And what's happening here, for the first time in our journey, we are having ADP getting converted to ATP. And since this reaction happens twice, we are going to have two molecules of ATP getting formed in this step. So what is this molecule going to be called? We had 1,3 bisphosphoglycerate, okay, and we removed the phosphate from 1, so this one is gone. And since this one is gone, the bis has to go as well, because there is only one phosphate group, and that's on the carbon number 3. So our molecule here is going to be called 3-phosphoglycerate. So we will look at this on the molecular level to see what is happening. As we saw here, we had our 1,3 bisphosphoglycerate, and there are two phosphate groups that we have. We're going to have the action of an enzyme that's going to be called phosphoglycerate kinase. This enzyme is going to remove this phosphate group from carbon number 1, and it's going to put it on ADP to produce one molecule of ATP. But since this reaction is happening twice, what we end up having is two molecules of ADP getting converted to two molecules of ATP. And what we have here, our new molecule, is going to be called 3-phosphoglycerate. And this is the first reaction in glycolysis which we are actually producing ATP. So far we have produced 2 ATP, but 2 ATP has been consumed as well. So far the net production of ATP stands to be 0. Now what we have here is that we have a molecule of 3-phosphoglycerate, which means our phosphate group again is on carbon number 3. This is carbon number 3, and our phosphate group is on it. That's why it's called 3-phosphoglycerate. So what we are going to do now, we are going to convert this into another molecule, preparing it for the reaction that comes after. So what happens here is action of an enzyme that's called phosphoglycerate mutase. And mutase are a group of enzymes that are going to move some functional groups on the molecule. So what's happening here, basically, phosphoglycerate mutase is going to remove our phosphate group from carbon number 3 and put it on carbon number 2. This is particularly important because in the next step we want to build a high energy phosphate bond on carbon number 2 to produce ATP from it. So what happens is that our phosphate group comes here, and this molecule is going to be called, as you have guessed it, 2-phosphoglycerate. And we're going to show you exactly what is happening on the molecular level for you to understand exactly what is happening. So what is happening here, we have our 3-phosphoglycerate here, and it's getting converted into 2-phosphoglycerate, and the enzyme that works on this is going to be called phosphoglycerate mutase. And what's happening here is that we have on carbon number 1, 2, and 3, our phosphate group here is on carbon number 3. And what's happening here, it's coming on carbon number 2, as you can see. And the name is going to be changed appropriately to 2-phosphoglycerate. And in the next reaction, we are going to have this bond being changed into a high energy phosphate bond through the action of enolase. Let's try to look at it first on our main glycolysis page. So a hydrolysis is happening here, and this reaction is going to create a high energy phosphate bond, on carbon number 2. And the enzyme that is working here is going to be called enolase. So our enzyme is going to be called enolase. And what it's doing is creating an enol phosphate bond, which we're going to see what it means exactly. And when this happens, it's going to be also a release of a water molecule from this reaction. So a water molecule is getting released here as well. So what is this new molecule is going to be called? Our new molecule is going to be called phosphoenol pyruvate. Our new molecule here is going to be called phosphoenol pyruvate. And that's because of the phosphate enol bond that we have made on this molecule. Let's try to look at its molecular form to see what is happening. So as you can see here, we have our 2-phosphoglycerate. We have our 2-phosphoglycerate here, and it's getting converted into phosphoenol pyruvate. Phosphoenol pyruvate. And which enzyme is catalyzing this reaction? It's going to be an enzyme called enolase. When you know what's happening on the molecular level, it's going to be much easier for you to remember the names. And as you can see here, we are making an enol phosphate bond. That's why the enzyme here is called enolase, and that's why the structure that we have here is called phosphoenol pyruvate, because we have a phosphate enol group. A phosphoenol is happening here because our phosphate is attached to the carbon backbone, which is connected to another carbon with a double bond. And as you can see, our hydrogen and our hydroxyl group has got removed here, and what we are seeing is in this reaction we have release of a molecule of water as well. So this is our reaction that we have made a high energy bond here again, and like the previous reactions, this high energy bond is going to be broken down to make ATP. So we reach our final reaction, which is the conversion of phosphoenol pyruvate into pyruvate. And this reaction is going to be catalyzed by an enzyme that we call pyruvate kinase. The enzyme is going to be called pyruvate kinase. And what is happening here is that our high energy phosphate bond that we have here is getting removed, and it's going to be put on, you guessed that, 1 ADP. And what we'll have is going to be 2 molecules of ATP, because this reaction, as we know, is happening twice. So 2 ADP to 2 ATP is getting generated here. And our final product, as we talked about at the beginning, is going to be pyruvate. And by this, we'll have covered all the reactions of glycolysis. So before we review it one more time, let's go and look at the final reaction on the molecular level to see what is happening exactly. So here we have our phosphoenol pyruvate, and there is a high phosphate bond that is here, as we talked about it in the previous reaction. So this reaction is happening through the action of pyruvate kinase, and we said that the kinase are enzymes that are removing a phosphate group and either from ADP or they're adding it to ADP to form ATP. So what's happening is that the phosphate group here is getting removed and is getting attached to ADP, and our ADP is getting converted to ATP. And again, as we said, this happens twice. So 2 molecules of ADP are getting converted to 2 molecules of ATP. So we have production of ATP again in this step. And our final product, as we all know, is going to be called pyruvate. And the fate of pyruvate is something that we talked about more in the video that comes about the TCA cycle. Now we're going to have a quick recap on what has happened so far. This is our pathway of glycolysis, as we talked about it in the video of overview of metabolism. And this is an important pathway because we want to metabolize glucose into pyruvate, and this is what it's happening. And these are the 10 steps that are involved in glycolysis. The first step, as we talked about, 1 ATP is getting consumed through the action of hexokinase. We are getting glucose 6-phosphate. Hexokinase gets its name. Hexo means 6. There are 6 carbon atoms, and kinase, they are taking phosphate from ATP and they are putting it here on carbon number 6, and that's why we have glucose 6-phosphate. And our glucose 6-phosphate undergoes isomerization, and what we have is going to be fructose 6-phosphate. Fructose 6-phosphate, through the action of a very important enzyme that's called phosphofructokinase, gets converted to fructose 1,6-bisphosphate. Why is it called that? Because we have 2 phosphate groups, one on carbon number 1 and one on carbon number 6, and that's why it's called 1,6-bisphosphate. Why do we call it bisphosphate? Because the phosphate groups are far apart from each other. This step is the second step that we are using ATP. Our ATP is getting hydrolyzed to ADP and the phosphate group is coming here. The next step is going to be an aldol cleavage, which is catalyzed by aldolase. This molecule is getting cut into half. We are going to have dihydroxyacetone phosphate, and we are going to have glyceraldehyde 3-phosphate. And what happens next is that our DHAP gets converted into glyceraldehyde 3-phosphate through action of an enzyme that's called triose phosphate isomerase. So by here, we are going to have 2 mole of glyceraldehyde 3-phosphate that has been made from 1 mole of glucose. From here onwards, each reaction is going to happen twice because now we have 2 of this molecule. The next step is going to involve an enzyme that is the dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase. Dehydrogenase is an enzyme that is catalyzing a redox reaction, which means our glyceraldehyde 3-phosphate is getting oxidized In other words, it's losing electrons, and these electrons are getting transferred to NAD+, which is getting reduced, or it's gaining electron and it's getting converted into NADH. There is an addition of inorganic phosphate that gets added here as well, and the molecule that we end up with is 1,3-bisphosphoglycerate. Now, in the next reaction, we have an example of something that we call substrate level phosphorylation. Substrate level phosphorylation. We are getting production of ATP without going into the electron transport chain. So what's happening here is that our 1,3-bisphosphoglycerate that had two phosphate groups on carbon number 1 and on carbon number 3 is getting converted to 3-phosphoglycerate, and this phosphate on the carbon number 1 is going away. It's getting transferred to ADP to make ATP. And this is going to happen through the action of phosphoglycerate kinase. And as we said, kinases are the enzymes that are going to move phosphate group into ATP. What happens next is through the action of phosphoglycerate mutase, we are going to be removing this phosphate group and putting it on carbon number 2. And the new molecule is going to be called 2-phosphoglycerate. And after this, through the action of enolase, a high energy enol bond is going to be made, and one molecule of water is going to get released. The molecule that we have is going to be called phosphoenol pyruvate. Phosphoenol pyruvate has a phosphate group on carbon number 2 that gets removed through the action of pyruvate kinase. This phosphate group is going to go on ADP to make 2 molecules of ATP, and we will be left with pyruvate. Thank you for watching the video. If this content is helpful for you, please subscribe to our channel. In the next video, we're going to talk about the regulation of glycolysis, and then we will talk about the TCA cycle and the electron transport chain.